My name is Helen Berman, and I'm Professor Emerita of Chemistry and Chemical Biology at Rutgers University. I've been involved in one way or the other with the concept and the implementation of the Protein Data Bank for the last 50 years. And from 1999 until 2014, I was the director of the RCSB PDB. The course that we're about to start is called Enabling Data Science and Structural Biology. And in this introductory lecture, I'm going to talk about the history of the PDB, and I'm going to talk about the various aspects of data science in setting up the PDB. In the rest of the course, there will be members of the RCSB PDB who will be giving lectures. And this particular lecture is an overview which will cover pretty much all aspects, and you may see some of these same slides later on as we go through the course. So I'm going to start now, and I'm going to tell you about the history of the PDB. In this first lecture, I'm going to talk about the history. I'm going to talk about the synergies of science, technology, and community. I'm going to describe the data pipeline and what we mean by that. I'm going to talk about the PDB and its usage, and finally about sustainability. And as we get into this, you'll understand more about what I mean about sustainability. The pioneers for structural biology were J.D. Bernal, Dorothy Hodgkin, John Kendrew, and Max Perutz. J.D. Bernal was a brilliant scientist at Cambridge and was referred to as the sage, and he was the mentor for Dorothy Crowfoot Hodgkin, who was a student there and was the first person to crystallize a protein and get X-ray diffraction pictures, and that was a landmark experiment. John Kendrew and Max Perutz, also in Cambridge, were the people who determined the first crystal structure of proteins, John Kendrew of myoglobin and Max Perutz of hemoglobin. And they were really the very first people to understand how important it was going to be to have structures of biological molecules. The early structures are shown here, and I have different depictions for each of them because there are different ways of representing atoms and molecules. And the first is myoglobin was the first structure determined by Kendrew. It's shown as a space-filling picture. Hemoglobin was determined by Perutz and is shown in this particular cartoon that has been developed for the use of representing protein structures. The structure of lysozyme was determined in the Phillips lab, also in England, and this is in Oxford. And this particular picture was a hand-drawn picture by Irving Geis. And then finally, the very first American structure, ribonuclease. All the others, all the work had been done for the others in England. And then finally, in 1967, we have a structure of ribonuclease done by Cartha and his group, and Fred Richards and his group in Yale. And I remember very clearly going to a lecture in 1967 in Atlanta, Georgia, hearing about this particular structure from these two groups and being so excited that we were finally in the business of protein crystallography in America. Okay, so these structures were being determined, and there were several people who really wanted to be able to do something with these structures, and they felt there was really gold in the structures, and if we could only have the coordinates, then we'd be able to do what we now call data mining. That word did not exist back in the 60s and 70s. So we had various kinds of meetings and discussions, and you'll see here a picture of a petition that we signed saying that there needed to be some kind of an archive for these structures. And please remember, at that point, there were maybe 2 handfuls of structures that existed, but we thought they were important, and so we pursued this. In 1971, there was a landmark meeting at Cold Spring Harbor in Long Island, and Walter Hamilton came down from Brookhaven National Laboratory and basically crashed the meeting. And at the end of the day in the bar, several of us came to see Walter and said, we really need to have a protein data bank. And he said, fine, I'll set it up. And that's how it began at Brookhaven. And the work was done by Walter and by Edgar Meyer, who had set up what he called a protein library. The picture at the top shows you a picture of Walter and Wyckoff, who was one of the people who determined the structure of ribonuclease. And then on the bottom is a picture of Walter and myself and Tom Ketzel, who later became the head of the PDB at Brookhaven after Walter's death in 1973. That meeting was in 1971 in June, and by October there was an article in Nature New Biology announcing the PDB, and I think importantly, when the announcement included the fact that it would be a collaboration with Olga Kennard in England, who was heading up the Cambridge Structural Database. So from the beginning, the PDB was an international project. And here's a nice picture showing all the luminaries who were at this meeting in Cold Spring Harbor, and basically the picture at the top shows you very, very typical people sitting in circle, talking about structures, talking about the exciting new era of protein crystallography. After the PDB was set up, there was no requirement that the data actually be put in the PDB. And there were several people who felt very, very, very strongly that the data must be there for this to be a successful project and to do good science. And so there were letters written to the NIH. There was another petition that was written, and this one was headed up by Fred Richards from Yale, in which he encouraged his colleagues to put their structures, put their coordinates in the PDB. And there were a couple hundred people who signed that petition. There were people, one of them was Dick Dickerson, who at the time was at Caltech and then UCLA, talking about that it was a scandal and immoral that the data were not in the PDB. And another person that I think was very important in all this was Marvin Kastman at the NIH, who was quite concerned that they would be putting money into health-related research, specifically AIDS research, and if the data were not available, it would not promote the development of drugs and so on. So there were, the journals got involved, the professional societies got involved. There was a committee set up by the International Union of Crystallography to come up with a set of standards for how exactly you would put data into the PDB, and more importantly, what data should be put in. What do we mean by that? And so in 1989, there was a article written in which it set out the exact guidelines. And this, again, was done by cooperative community action. It took probably about 7 or 8 years to make this happen, but it was very important because by the time it happened, everybody had bought into this. The other issue that was very important was the issue of data standards. How exactly do you represent the data? And how would you maximize the effect of having the data in this PDB in a way that you could actually get useful information out? So the International Union of Crystallography set up a committee to create these definitions for the structure and the experimental data from X-ray crystallographic experiments. And on that committee, the committee was headed up by Paula Fitzgerald. On that committee, in addition to myself, was John Westbrook, who is at Rutgers, Phil Bourne, who's the current Associate Director for Data Science at NIH. At the time, he was at Columbia. Sid Hall, who was the inventor of CIF, the crystallographic information format for small molecules, and Shoshana Vodak, who was a prominent European scientist who also knew how important it was going to be to have these standards. So over a period of many years, the definitions were set up, and by 1996, we had the first dictionary of terms to describe the X-ray crystallographic experiments. And we would have hoped that people would adopt it immediately, but people don't change that easily. So the PDB adopted MMSIF as its underlying format, but really nobody else did. And as the PDB grew, it was clear that we needed a much better format than what had been used before, and that we needed this MMSIF, which we now call the PDBX MMSIF. And that we had a meeting in England at the EBI. We brought together the software developers for X-ray structures, and to our great surprise and delight, they accepted PDBX MMSIF as the master format for the PDB. So it took a long time from the beginning in 1993 to the time it was actually accepted was 2011. The other issue that came up is the issue of the international nature of the PDB. And if you remember, when it was first set up, it was set up as an international project, but there was really no memorandum of understanding or no rules for how this was going to be run. And so in 2003, we established something called the Worldwide PDB. And the point of that organization is to ensure that the data are freely and globally available. We agreed that we would collaborate on all aspects of data processing and annotation and all the standards that were necessary for that to happen. And we also agreed that each person involved in the WWPDB could have different websites that have different services and different views of the data. So in that sense, it was more of a competition. So the members of the WWPDB are the RCSB PDB, which is in America, PDBE, which is in Hingston in England, PDBJ in Osaka, and BMRB, which is in Wisconsin. The other issue that became very important was that the PDB until 2008 contained coordinates of the model, and it contained what's called metadata, that is some descriptions of the model and some descriptions of the data, but it did not have the primary data. And many people said that in order to really be able to be sure that the structures that we have are correct, we had to have the structure factors. And so, again, a lot of community action, articles, people saying how important this was going to be. And in 2008, the WWPDB agreed that we would require the experimental structure factor data in the PDB. And that was announced to the public. Okay, so now we have the coordinates and we have the experimental data. So we can actually do what's called validation, and you will hear more in the course about validation. But basically, how do you know that the model that's in there is correct? How do you know it's the right model? And in order to do that, we had to have some kind of standard for validation. And again, we wanted community input to this, not just the PDB say, well, this is what we think, but rather find out what the experts think. So the very first X-ray validation task force was set up in 2008, and it was headed up by Randy Reed, who's at the University of Cambridge. It had 17 members, and these people went through the entire PDB at the time to try to figure out what is the best way of telling whether a structure is correct or not. They did a lot of different kinds of analyses, a lot of different kinds of tests. They ran all their tests through the entire PDB until they finally came up with recommendations for how exactly an X-ray structure should be checked and validated. And an article was then published in 2011 in the journal Structure. Shortly after the setup of the X-ray validation task force, we set up an NMR task force. And again, that was headed up by Guy Monteleone and Michael Nilgis. And then that group tried to figure out, how do you know whether an NMR structure is correct? And that group had many meetings and continues to have many meetings, has published a white paper in Structure with their recommendations for how NMR should be checked. The next set of recommendations were made by the Electron Microscopy Validation Task Force, which was set up in 2010, headed up by Richard Henderson and Andre Sali. And again, trying to figure out, how do you know whether or not an EM-derived model is correct? And so a set of recommendations were made. That work continues to go on, trying to actually figure out what the validation standard should be for EM, and that's a work in progress. Small angle scattering, another method which is used usually in conjunction with either NMR or EM. Jill Truella headed up that task force, and they came up with recommendations. And then finally, in the current world that we live in, structures are being determined by using multiple methods and a lot of computation, and we don't really have a clue how to check those structures or how to know whether they're right or wrong. So there's an ongoing effort in what's called hybrid methods or integrative methods, and that work is being headed up by Andre Sali, Torsten Schwede, and Jill Truella, who was working again with the community to try to figure out how, in fact, we'll be able to check those structures. So very active, lots of community involvement. It's really the only way you can be able to make the protein data bank or any data resource work properly, is to have all of this input. Okay, so I've shown you that there's a lot of different science involved in the PDB, a lot of technology, and I've emphasized, because I believe it to be very important, the importance of community. So I have a little graph here to show you the progress of the PDB over the years. First in the science, where at the very beginning there were just some small enzymes. There was tRNA in 1974, was determined. In the 1980s, we began to see the structures of small DNA fragments and then these gigantic viruses. In the 1990s, we began to see the protein nucleic acid complexes. when the RCSB PDB first became in charge of the PDB in 1999, one of the earliest structures that we got was a ribosome, and that was very, very challenging because it's a huge structure with both RNA and protein, and that was a difficult structure to process and put into the PDB. And now in this decade, we're worrying about these large macromolecular machines. In terms of technology, at the very beginning, we were only dealing with X-ray diffraction, diffractometers. The data were on punched cards, and there was compared to now, a very primitive technology. In the 1980s, we began to see synchrotrons, which would allow you to get extremely intense X-rays. Computer graphics began to be used, and then we began to also see structures determined by NMR. In the 90s, electron microscopy began to be used as a method to determine structures. And then we began to have very, very fast computers and very, very fast and accurate detectors. In the 2000s, we saw high throughput structural genomics where it was possible to use robots to get structures to be done very, very quickly, and I'm happy to say also very accurately. And then in 2000s, we're beginning to see 2010 and onward, we're beginning to see structures by integrative and hybrid methods. And here at the bottom are the progress of the community interaction, starting in the 70s, where the archive was established when there were just a few structures. We begin to see the importance of community involvement in setting up guidelines for deposition. In 1990s, we begin to see the importance of standardization and the birth of the RCSB PDB as a replacement for the protein data bank that had been at Brookhaven. And then in the 2000s, we see the groups from around the world coming together to form the WWPDB so we could have a cooperative effort. We see the requirement of experimental data as a way of seeing us be able to validate, and now in this current decade, we see a big, big emphasis on validation. So I come back to the beginning of our founders, and remember I told you that J.D. Bernal was responsible with Dorothy Hodgkin for getting the first diffraction pattern of a protein. But J.D. Bernal was a very interesting man and a key influence on how the crystallographic community works together. And he believed in the importance of social action, and he wrote a book on the social function of science. And he covers the organization of research to science and its social role, and made the statement, and he was a communist, that science is communism. I think the idea of people working together in communities is something that has stuck with structural biology till this very day. When I gave this lecture, or something like this lecture, many years ago to the librarians at Rutgers, they said to me, "Oh, the PDB is an example of the theories of Elinor Ostrom." Elinor Ostrom won a Nobel Prize in economics in 2009. And I didn't really know her work, and so I decided to find out, and I talked to my colleague Avi Dixit, who's at Princeton and also an economist, and he explained to me that what Elinor said was that there was bottom-up collective action can work better than top-down enforcement, and for sure the PDB has operated that way very effectively. And He pointed out that what she believed in is that the groups devise clear rules about what actions are and are not allowed. They're based on local conditions and knowledge. We have to be adaptable to changing circumstances and include organized monitoring and enforcement. And as I've told you a little bit, and you'll hear more, that's basically how the PDB operates. Okay, I've just given you a summary of the history of the PDB and tried to give you a sense of the way in which science and technology and community all work together to form this kind of an archive. And in the next lectures, I'm going to actually go into much more detail about how the data are collected into the archive.